Female infertility is a deeply emotional and complex issue affecting millions worldwide, and new genetic discoveries are reshaping how we understand this challenge. But here's where it gets controversial. A recent groundbreaking study has spotlighted a particular genetic variant in the JARID2 gene as a major player in female infertility, especially through its impact on early embryo development. This research, conducted by Sun and colleagues, dives into how a specific homozygous mutation in JARID2 severely disrupts blastulation efficiency, the critical stage where a fertilized egg transforms into a blastocyst ready to implant in the uterus. This finding doesn't just fill gaps in scientific understanding, but could radically change infertility diagnosis and treatment strategies. The urgency behind this research stems from the stark reality that 1 in 6 couples worldwide confront fertility struggles. While infertility has long been recognized as a multifaceted problem influenced by many factors, genetic causes are often the hardest to pin down. The focus on JARID2 introduces a vital genetic piece of the puzzle that helps explain why some embryos fail to progress beyond the early stages despite fertilization, leading to unsuccessful pregnancies. To unpack the significance, it's important to understand JARID2's role in our biology. This gene is key in regulating how other genes turn on and off during early development by modifying chromatin, the structure that packages DNA. When a harmful variant occurs in JARID2, the delicate orchestration of gene expression during early embryo formation falters. Sun et al. Carefully demonstrated that women carrying this homozygous mutation had embryos with markedly reduced ability to reach the blastocyst stage. Without this crucial developmental milestone, implantation and subsequent pregnancy become highly unlikely. The researchers combined multiple scientific techniques, including genetic sequencing, laboratory embryo culture, and detailed observation of developmental outcomes, to firmly link the JARID2 variant with blastulation problems. This wasn't just a theoretical association. The findings were both statistically and biologically convincing. This level of evidence elevates the study beyond simple correlation and opens a door toward practical applications, like genetic testing for patients experiencing unexplained infertility. And this is the part most people miss. By identifying such precise genetic factors, fertility care can move into the realm of personalized medicine. Instead of routine treatments applied broadly, fertility specialists might one day tailor interventions based on an individual's unique genetic landscape, drastically boosting success rates and potentially reducing the emotional and financial toll of repeated infertility treatments. The scientific community has responded enthusiastically to these findings, recognizing JARID2 as a promising target for further exploration. Experts in reproductive genetics are particularly keen to investigate how this gene interacts with others involved in early development, as this could reveal a complex network of genetic influences that previously went unrecognized. Could this mean new prognostic markers or novel therapies? The possibilities are vast. Beyond the genes themselves, the study invites broader reflection on how genetics intersects with environmental and lifestyle factors such as diet, stress, and exposure to toxins. These influences might exacerbate or mitigate genetic predispositions, pointing toward comprehensive, multifaceted strategies for improving fertility outcomes. What makes the story of JARID2 especially compelling is how it illustrates the profound effects tiny genetic changes can have on human health. It's a timely reminder that fertility isn't simply a matter of chance or lifestyle choices. It's deeply rooted in biology. This focus on the genetic dimension is crucial for advancing women's health research and highlights the critical need for ongoing investigation into reproductive biology's complexities. Clinicians stand to benefit significantly from this research. As genetic screening technologies become more accessible and sophisticated, identifying women carrying JARID2 variants could become a routine part of fertility evaluations. This shift could revolutionize treatment pathways, enabling earlier, more targeted interventions that improve outcomes and patient experience. At the same time, the expansion of genetic testing in fertility care raises important questions and concerns. How will patients navigate the ethical, privacy, and psychological implications of knowing their genetic risks? Will broader acceptance of genetic diagnostics lead to better informed, more empowered choices? Or might it create new anxieties and disparities in care? These are important debates that the medical community and society at large must engage with as this science moves forward. In summary, the study by Sun et al. Uncovering the impact of a homozygous JARID2 mutation on female infertility is not just a scientific milestone. It's a beacon of hope for countless individuals struggling to conceive. This discovery deepens our understanding of the genetic roots of infertility and points toward more precise, effective approaches to treatment. As research progresses, integrating these genetic insights with compassionate care and patient education will be vital to transforming fertility healthcare. The path to resolving infertility through genetics is intricate and evolving. But the revelations about JARID2 mark a pivotal step. By combining cutting-edge science with practical applications, the future holds great promise for changing fertility treatment from a frustrating guessing game into a more predictable, personalized journey towards parenthood. What do you think? Should genetic testing become a standard part of fertility assessments, or do concerns about ethics and accessibility suggest caution? Share your thoughts and join the conversation.